I'm switching gears. I'm going to go a little bit right-brained. I hope I'll be flipping back and forth. I just want to share with you an email I received last night from one of the students who is working with the blocks. Not one of the three that I talked to you about, but yet a next generation of block projects. And it says, "Hi Jane, sorry, I'm going to get choked up. I worked myself into a lather, and I'm not choked up about this letter, although it's kind of nice." Sorry, not lights yet, sorry Clive. Well, it looks like my CCO is our college, research into feedback and palpation may open an interesting door. I am planning to meet with a professor at McMaster, which is a nearby university, regarding the possibility of doing a neuroscience PhD on multisensory integration amongst experienced osteopathic practitioners. That's what we're here for, to try and encourage this, right? They have expressed preliminary interest in the topic. Specifically, I would like to do fMRI work, functional fMRI work, to examine the details where in the cortex osteopaths are processing their tactile information. Based on the theories of Baccaridi in the area of neuroplasticity, along with the hints about visual and auditory processing of palpation we've heard from some of the instructors at the college, tactile processing must be spilling over into other areas of the brain. I want to see for myself and to quantify it. In your travels, have you heard of anything similar being done? No. Since I'm traveling, no? Okay, thank you. Would you be open to talking it over sometime between now and January? Yeah, put in my calendar. I know it's not CCO related, but I'm excited about the topic, and I'd be thrilled to have any input you have time to give. If I can sell them on the research, it would likely have a start date in fall of 2015. I'm crossing my fingers. Thank you for all your hard work you are putting into the, oh, that's something else. It's another hat I wear. Oh, isn't that great? So, experienced osteopathic practitioners may be doing a road trip. I'll plan some sort of conference in 2015, and maybe we can work this together. I'm going to talk about qualitative research. And just as I get started, is John Lewis in the audience? John Lewis. Sorry, I haven't met you yet. I have a letter from one of our researchers in Canada. That's what I do, I deliver. Yes, it means work, of course it does. So as the same as yesterday, I'm using the same slide that I'm going to take you on a bit of a circuitous route to get where I'm going. So I'll talk about qualitative research, beginning with what I didn't do. What I did do, and my experience from qualitative research, which is what Clive asked us to talk about. So the big deal with qualitative research is, so what? Any kind of research, who cares? So I'm interested in something, yeah, but so what? So I give this to the students all the time, and they're a bit perplexed at first, but they have to dig into it to see why their topic is important. If you take time to look at this little slide here. Hi, I'm a graduate student, and I'm learning about a new type of research that focuses on the lived experience of different life events. I just have a few questions. There's like this one, phenomenological research. Maybe a little bit inappropriate. But I think the beauty of qualitative research is that it's hypothesis generating for those of you who want to conduct the quantitative research, when you don't know where to start. Because it examines concepts and phenomena that are not measurable, that are intangible, that can't be validated in a laboratory, such as what is the tide? transmutation, the V-spread, fulcrum, bioplasm, biogen, vitality, potency, still point, intent, palpation, the dura, and the cranial, the three cranial vertebrae. Those are some of the topics. I don't think anyone's conducted the tide. I'm working to get someone here to work on the V-spread, but these are all topics that have been taken on by students at our college or the Montreal College, and I think I even supervised somebody from the ESO on the Stills concepts of biogen. So imagine just taking one word from an entire book and trying to do a thesis on it. Sutherland's concept of transmutation, and writing a 200-page thesis on it. Find out what it is, because before you can actually do an experiment on it, you have to know what it is. So as an example of the V-spread, do you teach it here? Mm-hmm. I've seen it taught so many different ways. And basically you sort of, right? And you've got a physics professor there lying on your table saying, what are you doing? I'm sending you fluid. You have the obligation to be able to explain what you're doing. So I found this explanation. The impulse is directed straight through the contents of the skulls and sutures, and force is transmitted along the skull's diameter to facilitate movement in the sutures. Those are two completely different approaches. And what's worst of all, is osteopaths are teaching other professions some quick osteopathic tricks. So you have physiotherapists and massage therapists and sports therapists on the rugby pitch going for a concussion, right? So it's not a joke, it's happening. So these are some questions that you would ask when you conduct a research topic, and I've put on some bad answers here. What are you going to do? I want to do the V-spread. Why are you going to do it? To understand it? Why are you going to, how are you going to do it? Read all the stuff about the V-spread and then try and put it all together? And so what? Then we'll know what it is? And that's how it starts. And then I look to Viola Fryman for help here. She really distinguishes between an essay and a thesis. An essay is just gather information and try and synthesize it together, but a thesis, you give you a pound of flesh. Something comes from you into that paper. So you have this in your handouts, but the ultimate objective of a thesis is for you to reveal how you think, how you reason, how you deliberate from one area to the other. It's not good enough to be a documentation of what other people have written, no matter how far back you may go to get it. That's only information. What we want when we judge a thesis is what your thinking process is. How have you digested all this stuff that you put together, and what have you come out with? What's it all mean? And those are her words echoing in my ear. So here's a better justification for the student. Since information on the V-spread in the literature is scant, and much of the available literature contradicts itself, and students seem to find the concept elusive, and patients ask, what are you doing? That's a reason, that's a justification for conducting a thesis on the V-spread. And you only have to know that law where, if you think left brain, there's a law of physics where if you push on the outside of a balloon, pressure is exerted equally in all directions. So here you are doing this little, and you've got a 15-pound head leaning right on the table. So you're trying to overcome this 15 pounds of already force from the table with your little finger. So anybody who wants to participate, who's got some insights into V-spread, please contact me, because the woman who's going to engage in this project could really benefit from listening to others outside of our college. So she's picked out a whole array of college people, but they all learn from the same master, which doesn't help. So the questions might...
Jane, so we usually pair the idea of a V-spread with fluid drive, so we call it the V-spread and fluid drive, and that kind of slightly changes the mental picture. Do you
Yeah, and
use that term?
she listed a whole bunch of stuff she wants to find about fluid dynamics. So she's prepping it all that, and then going into the...
And when you teach it, do you just call it the V-spread?
Oh God, I don't teach it. No, it's just a V-spread. Oh no, what we're taught now, if I remember, first of all, you find diagonally opposite sutures. From this side, you send an impulse to that side to make sure you're there. You feel heat coming into your hand. And then you reverse it, and you just send the fluid. And some teachers...
So that's the part that's the hardest to explain to anybody. The V-spread is kind of, well, relatively
And then I've
mechanical.
seen them argue at academic councils whether you V-spread. No, you have to mold your fingers to the suture. So you V-spread. Sorry, drives me nuts. But as an osteopathic historian, I can tell you that I've been to Texas, I've been to the archives of the Cranial Academy, which they got hidden in boxes in a room that nobody knows about, and I've read all the private letters of Sutherland and Roland Becker that they have there, and Roland Becker said it is the most accurate way to diagnose a lesion in the cranium is with a fluid drive. But we don't know how it works. Those are his words. It's in my filing cabinet at home. That's a justification for doing a qualitative thesis. How do you find that out? You have to ask me if I know anything. I know stuff. I just don't know what I know. So you have to ask to find out if I know it. I can't catalog it. Do you see where my passion is now compared to yesterday? Yeah. And I haven't even got to the fascia yet. So how is the V-spread concept taught and understood by osteopathic practitioners? In light of 21st century science, can a deeper understanding of the concept of the actual mechanism be understood? That's a better set of research questions. And how can the information from questions 1, 2, and 3 be synthesized to help advance the osteopathic concept of the V-spread? This is the role of the tutor or the thesis supervisor that comes in to help shape the osteopath, the student's thinking. And these are bounced around, and they usually have 7 questions, and you can get them down to 6. And I know how much work it was. 3 or 4 questions is ample. So now we'll get to what I did do, which was Still's concepts of fascia. These slides are going to come as a surprise to me, because I had a meltdown last night and didn't look at anything since I left Canada. I'm revigorated right now. So I chose this topic because if you start to read the chapter on Still, chapter 10 in Philosophy of Osteopathy, you're just bombarded with these words you've never heard of. So I know in the first page they talk about the quickening atom, the active principle, the vital germ. Something I took in my fascia course. Nothing I ever heard those terms before. Where did they come from? What did they mean in that time, in his era? Those were all problems that I had to solve before I could even come close to figuring out what he meant. So I asked the questions, what were Still's concepts of fascia? Then I complicated my life by asking modern day osteopaths what their concepts of fascia were. And then I asked them what their understanding of Still's concepts of fascia were. And then I asked if there was any difference between, especially, well, all 3 of them. What's the difference? And So what? So what? So the method was a historical documentary and field work. And it was literature-based and interview-based. Now, there's no peer-reviewed literature on it, still per se, but I looked at everything he wrote, everything that was written about him, everything that he read or that I knew he read, and so on and so on and so on, and the whole 19th century, sorry, 18th century America with Swedenborgianism and the New England Chinese, New England Immigrant Society, there's at least 20 intellectual currents, and probably John found a lot more. But you had to get into that field to understand what he was talking about. In the data analysis, I used a template design, which meant I took my data with no preconceived ideas of what I was expecting to find and tried to sort it into themes. There's three kinds of forms, but that's the one I use, and I think I was right in using that one. The synthesis, I used immersion crystallization, which really just means sit with your data forever, let it drive you crazy, live it, breathe it, regurgitate it, and something will come out of it. And in fact, it wasn't until I was walking my dog with another girl, and I'll tell you that when I get to what Still's concepts of fascia mean, when it all crystallized for me. And it happens for everyone who uses this method, is if you live with that, the idea will come and you have to force nothing, it's all there for you. So, sorry here, with how did I find out his concepts? I can tell you in 10 minutes, or I can tell you in 4 days. So I have to give you the very, very short version. I looked at his concepts of fascia within his paradigm of thinking, maybe not philosophy is not the best word, within how he conceived osteopathy and with America in the 1800s. And one of the things he did as a young lad was hunt. And he hunted for food and clothing for his family. And he started killing deer at 8 years old, before he owned a dissection book. Before someone told him that you have to cut away the fascia to see the underlying bones and muscles and nerves and arteries and veins. He has that book on his shelf, a Dublin dissection, but he didn't have it when he was 8 years old. So he saw what he saw, and no one told him not to look at fascia. And he saw the stuff and the stuff. So the short of it is, when I put all that together, I can distill. Stills, they don't get that in any other language. His concepts of fascia down into two things. That he conceived it as a membrane and as a system. Now, the membrane I got when I was walking the dog with this other girl. And I said, "You're doing your master's, aren't you?" And she said, "Yes." And I said, "What are you doing it in?" And she said, "Osmotic forces and membranes." And I heard nothing else she said the rest of the way. I just heard membranes. And that was the piece I was missing all along because I was looking for fascia. I was trying to figure out fascia. It's membranes. Shit. And the system, again, another long story that I don't have time to get into, but it's really from complexity systems. He was talking in a language and conceiving of ideas that hadn't been invented yet. He was 100 years ahead of his time, and he was speaking to an audience that only thought linearly, and he was in a non-linear way of thinking. Yeah, skip him. Vichy was an anatomist who was actually written about in Mueller's book of physiology, and still owned a copy of Mueller's book. Like everything in the United States that you want, somewhere along the line, it was in a fire. whether it's a post office or a records office or somebody's attic, everything gets burned in the United States. So most of, there's only about 20 books left that we know he owned and read. Mueller is one of them, and in it was Bichet, is a French anatomist, died at the age of 31. Bichat.
Yeah.
Sorry.
I can give you a copy of his Traité des Membranes.
I have.
Oh, you
I
have?
have, of course.
That's amazing.
Yes, thank you. So Bichet classified them as fibrous, serous, and mucous membranes. That was the tissues of the body, or combinations of, so fibrous, serous, mucous membranes. He did it all without a microscope. That was his classification. Our classification is connective tissue epithelial. He just mixed up these terms, membrane and fascia. He interchanged them all the way through his books. So when you see something like the fascia proves to be the probable matrix of life and death, you can substitute that for membrane as well. He said it, in some places he said, it doesn't even matter what you call it. Call it fascia membrane. He didn't say call it fascia membrane, but he meant you can call it fascia membrane. So this connective tissue are these two, and the epithelial and the mucus are the same tissue. This guy, Eric Bleschmidt, who studies his embryology, anybody here? You people, yeah? This is what's interesting because Bleschmidt sees these as inner tissues, and this is the frontier tissue. It's still consistent even with human embryology. As for the system, he looked at flows. And I can only describe it this way. If we have a system here, and flows going through the fascia, or fluid able to permeate through the fascia, and we put an impediment at one side, you're going to get this shrinking. Whites actually live, whites fail to shrink or swell and die. And likewise, if you put the impediment at the other end, you get a swelling. Now, he said pretty much the same thing about the diaphragm, right? Whites actually live and whites fail to die. But you have to understand about still, he just got out of bed one morning and wrote on something. Everything was important. He wrote more about earwax than he did about fascia. And that's why you can't isolate one area of his work and say that it was more important than anything else. It's not the case. I just happen to focus on fascia. The results from questions 2, 3, and 4. So this was interview-based. I used a convenience sample and a snowball sample. So convenience means who's around and who's going to talk to this Jane Stark chick. And snowball means, now that you've spoken to me, can you recommend someone else I could speak to who actually knows about this topic? So I interviewed 37 osteopaths or osteopathic physicians, and I'm proud to say 2 of them are in this room. Stuart, I don't know if you remember.
I do.
Yes, and Ernest also took part in this. There's always one of you in my audience, which makes it a little more difficult to pull off the conclusions. So Viola Fryman was actually on my jury. No pressure there. And Michael Patterson. Okay, so they each had 20 years experience in a manual-based practice. Together they had over 1,200 years of experience. And I sought their consent not only, well, I sought their consent, they gave it to me. And I did an unstructured interview design, which means I had a set of questions, but I could alter my order of them if they said, I told you I didn't do that. I didn't have to ask the next question, which says, how do you do that? Right? It allowed me to listen to what they were saying. And I asked them a crazy number of questions, 20, which is way too many. And most of them were open-ended, so thank you for putting up with me. I won't foist any students on you with 20 questions anymore. The inquiry took 3 areas of approach. How familiar were they with still? What's their concepts of fascia? And what's their understanding of still's concepts of fascia? So once I had the information from the 37 interviews, instead of analyzing it by person, I analyzed it across the top by question. So I took 1 question, and with that template design, I tried to pull out common factors. So I usually got 4 or 5 sort of themes I saw, and then I made a discussion on the paper about the theme. I linked the themes together and came up with a conclusion on 1 question. So what's the overall conclusion on this question? And I did that all the way up to 20 questions. So then I took the conclusions from all 20 questions and tried to make a final conclusion. It took me 900 pages. And that is that each osteopath has an individual conceptualization of fascia. But that's enormous, because each one of those is going to be teaching you a class. Okay? It's their concept. It's not the law, it's not the rule, it's not how it works, it's just the way it happens to be for that individual. The answer to research question 3, which was, is each osteopath understands Still's concepts of fascia differently, or don't understand Still's concepts of fascia. Now for research question 4, how does modern day osteopaths' concepts of fascia compare to Still's concepts of fascia? I had to interview Andrew Taylor Still. Yes, that's me. So I asked Andrew Taylor Still the same questions that I was asking the osteopaths, and I went through his work and I tried to find the answers, and then I compared them to what the osteopaths did. That was fun. And then I made a conclusion for all the questions, then I linked all of the conclusions together. And I think this, being able to do this has helped me make maple syrup. Because if you know anything about collecting sap, you have to boil it down from 50 gallons to make 1 gallon. This is really what I did with this information here. So is there a difference between how the osteopaths have still understood fascia and how the osteopaths understand still? Yes. So I'm going to give you a question and what AT Still would say and what the osteopaths would say. Do you routinely assess and treat your patient's fascia? No. Yes. What's your goal when you treat the fascia? I don't. But I like to ensure that there's good fluid flow. It's always concentrating on the fluids. Osteopaths did not mention fluids. It was, now I had 21 Americans, 9 British, what's left? Some French, maybe 6 French. And I don't know if it was the nature of the question, but transcripts are readable, and you'll find out there's virtually no mention of flow. There's 3, but they're not in the same concept as still do. And then how do you know when your fascial treatment is successful? There's fluid flowing. And they didn't mention flows again. They talked about color, they talked about symmetry, they talked about decrease of tension, they talked about their hands not feeling anything, but the concept of flow was not there. So why did I think there was such a difference? Why? When you look at their familiarity of still, their education on still is very limited. An hour lecture, 2-hour lecture. They were introduced to him through quotes and one-liners, and they still give out that stupid book by Webster, The Sage Sayings. There's one-line quotes, John. They still give out that at graduation from osteopathic schools. That's what they know still by. They were either self-taught or not taught. The books by Still, there was no translations. They were hard to find, hard to read, and hard to understand, or not read. So implications for the research. I don't know what that meant. Oh, this is my summary, but I'm not finished yet. Because then I'm going to tell you what all this means to Nicole. This was just to say the summary was still conceived of the fascia as a system and as a membrane, and that he mixed up these fascia and membrane. But the question is, if each osteopath has an individual conceptualization of fascia, and each osteopath understands Still's fascia differently, how do we teach fascia? From a body of knowledge, if you say body of knowledge, or from an individual basis. And if you say body of knowledge, I ask you, what's the body of knowledge? And I ask, and is it an individual basis? If it's an individual basis, what unifies osteopathy? Bruno gave you the answer yesterday, who remembers? Who's that? What did Renzo say? Hey, are we linked?
Yeah.
Yay. Unity in diversity. And we're getting a body of knowledge. We have now 3 fascial research congresses. The information on fascia is fast expanding. But so what? Here's the so what. I got this letter after I finished I presented in November of 2003. I got a letter in July 16th that says, thank you very much for the copy of your thesis. You have made a superb, in-depth study of the philosophical foundation of osteopathy, gaining access to much literature that I'm sure was unfamiliar to many of us. May I have your permission to quote from this from time to time and even share it with my students? I congratulate you on excellent contribution to osteopathy literature with all good wishes as you move forward in your practice. May God bless you day by day. That's a good so what. She flew in, literally was given the 900 pages, read the abstract, and then she took it home and wrote the thing. I don't know how she got it home, but she did. All right, so what? What has happened? How has it changed my practice? First of all, I was hunted down for a year until I published it. And it came out in German first, and then in English. I haven't slept since the day I presented. I never intended for this to happen. I have been running ragged. I'm now on the faculty at 4 campuses of our college. I've given workshops on fascia, treating fascia. I'm number 38 with another idea, for in 10 countries, including Kirksville. They invited me to come down and teach them about, it wasn't actually about fascia, it was about Still's approach to the body, which included fascia. I'm director of research at the CCO. I write at least 1 article per year. I've done 2 book chapters. Upcoming, I'm going to be co-editor for an osteopathic text. I've been asked to do 2 more chapters on Still. I've got 3 books on my desk that I haven't finished. I just joined the faculty at the Japanese college, and I'm taking 3 postgraduate courses concurrently. Now, What else? And remember, I started 5 minutes late.
And you do maple syrup.
And I do maple syrup. And this is my backyard as my clinic window. It's a little bit of a close-up, I must admit. But that's Bambi. That's my husband. I guess Bambi's dad. And that's me. That's my workshop. And these are the guys that work for me. This is the one I push around all the time. That's me right outside my backyard, right outside my clinic window. Fresh kill, into the meat, into the fascia. What does it look like? Yes, it stopped breathing, but it's alive. Just a few representative samples. If you look for it, you can find fibrous, straight, parallel fibers of fascia. But mostly you don't find that anymore. You find layers upon layers upon layers upon layers of this thin fascia. You can't stretch this stuff. I don't know what we're doing when we're doing this, but you can't stretch it. You can bring it to length, but you can't stretch it. All it does is distort from somewhere else. You know what I mean? If you pull it this way, it'll distort another way. This has got to stop. This animal here, he's already 3 days old, I think, but this is his back. This is a cadaver back. This is what they do from the cadaver back to make the pictures that we study from our textbooks. The textbooks are wrong. It doesn't look like that. We've got the wrong mental picture of what the fascia is like. This is the diaphragm about half an hour after the animal's been killed. Looking up. This is the most beautiful piece of tissue. This is the mesentery. It's like stained glass window. And you know that the cell membranes are cell membranes because they're in environment of water, right? Because of the bipolar lipid that turns itself hydrophobic. But without the water, there's no cell membrane. And if you hold that another couple of minutes, it'll just disintegrate on you. And you'll just be left with the fat and the arteries. It's fascinating. You need the water. You have to have the water. So when we were studying inner tissue, the connective tissue, the fibers or the serous membranes, the flow between the layers is what's important. I don't concentrate on the fabric anymore. Concentrate on the fluid. Because within the fluid, you have the possibility for every chemical reaction in the body. All the defense systems, the immunes, the inflammatory, the nervous reaction, everything happens in a fluid environment. And that's teacher number 38, teaching about the fluid approach to the fascia. Oh, because embryologically, first comes the fluid, then the matrix, then the cells, and the cells as a byproduct of their cellular metabolism spits collagen into its extracellular environment, which makes the collagen. It's the last thing that comes through from embryological process. Just a couple more. I've added no water to this at all. That's the deer here that you see here. I think some of the bubbles are just artifact from pulling the layers apart. sorry, it's too bright in here to see the vessels, but here's one that's blue, a tiny little vessel in the superficial fascia. That's what it looks like, this, Bruno, there's no order to it, but without the order, you can move in every direction with it. It forms and reforms, forms and reforms, forms and reforms, but not without the fluid. Good, eh?
Mm-hmm.
So I'll leave you with a quote from you. I leave you to study and practice the philosophy of osteopathy as here set forth, governing yourselves accordingly and forming conclusions of your own based upon the day-by-days unfolding of the science.
So, do we have any questions?
Have you published, or do you intend to publish any of those photographs of the fascia, those fresh?
Oh, I never thought of that, no.
They're some of them are very geological. You showed a sort of image which could have been marshland or sort of research on earth sciences. You'd find such similar images.
No, I never thought of it. Sorry. I'll add it to my list.
Please do. Yes.
One of the problems I can see with doing interviews with osteopaths is that we don't have a common language. So some of the differences may be because of use of language rather than actual philosophical differences.
And that's important, and that's why the qualitative research on something like vitality, where she interviewed osteopaths asking them what vitality was, trying to come up with a common definition, yet of course they sit on the shelves unpublished. But we're doing that step by step with all these terms, so that when Stuart talks about vitality and we talk about vitality, we're talking about the same thing, but she found it was really like the elephant in the room. So they were talking about vitality, but they had a different part of it. And that made it hard. And there's lots, as a student, there's lots of mistakes I made. My pilot testing was done in the elevator with my advisor going from the basement to the 4th floor the night before the first interview. I pilot tested the questions. When you say, how do you know when you're touching the fascia? Well, I didn't describe what touching is. Nobody asked me, what do you mean by touching, which kind of surprised me. No one asked me for clarification, but that's open for all kinds of interpretation. How do you interpret what I mean by touching? And I got answers like, well, every time you touch a person, you're touching the fascia. You can't touch the fascia, only the surgeon can touch the fascia, and anyone who tells you can touch the fascia, they're blowing smoke. That's probably a mistake of the question, or not.
Yeah. Sorry.
Marcia?
Hi. I wanted to say, I believe that Nottingham University, they did some work on how acupuncture works. And one of the things that they came up with, I believe, was that it works via the connective tissue matrix using piezoelectric lines of force.
No, I don't have a problem with that at all.
No. But that's what fascinated me, that idea that you've got this crystalline piezoelectric force field, as it were, and also the idea that you've got Langer lines of stretch, also the idea that you've got that the tissue follows lines of stretch.
Okay. The tissue is formed as we grow. So when you look at embryology, is how did we develop from the embryo? Because the fibers are going to line up in the line of stress at that particular time. right? So you can see in the directions, when you see so many layers, you know, oh, he grew, he grew, he grew, he grew, he grew this way, he grew that way, he grew this way. I mean, right here, we have at least 12 major layers of fascia. And you take a cesarean section, you cut through all those layers, and then you zip it all together with one stitch, and it goes this way, that way, that way, that way. Well, like this.
One question.
I'm going to leave it actually now.
Do you see any benefit in stretching fascia at all?
Pardon me?
Do you see any benefit in stretching fascia at all?
Nope. No, because you want water in it. You don't want to wring water out of it. I try and float one layer on the next. We're the incompetents that need to name it. It doesn't know it has a name. It is one tissue. And we compartmentalize it. It doesn't stop and start. It was formed as a continuity. And it's not just a continuity because it grows another layer and another layer. It's also contiguous, one on top of the other. And it's the contiguity that becomes the problem where we don't get the sliding one over the other, over the other, over the other. Where's Bruno again? In your aquarium in the sea where all those anemones are moving back and forth. That's how we have to move. And those tiny, they're about 30 microns thick layers of fascia with 43 microns fluid between those 30 micron layers of fascia. See how much fluid is there? Thank you very much.